नमस्ते हमेशा खुश रहो बिटिया I am JVN Dr. Lily Trivedi and today I am going to take before you diseases of groundnut. Now in this disease we are going to discuss the early blight, hortica disease of groundnut and the late blight and also the wilt disease of groundnut. Now we are having in the early leaf spot is caused earlier known as ticca disease of groundnut is caused due to Cercospora archidicola, whose sexual stage is Mycospirilla aciridis, and the late leaf spot is caused due to Phaseosporiopsis personata. Another name is Cercospora personata, and the sexual stage is Mycospirilla burkilli. Now, what are the symptoms? Now, when we come to see the symptoms, the disease is produced on all the above ground parts with more or less severe on the leaves. Now, the leaf symptoms are produced by the two pathogens and they can be easily distinguished upon the spot color and shape. Both the fungi produce lesions on the petiole, stem and pegs. The lesions caused by both species coalesce together and as the infection develops, the severely spotted leaves shed premature. So the quality of nuts and the yield is drastically reduced. So here you can see this is the early leaf spot and this is the late leaf spot. Cercospora archidicola and Cercospora personata. Here's another picture showing the distinguishing between the two. Early leaf spot and late. And here as it progresses, it destroys the whole leaf and premature falling of leaf takes place. The pathogen is now intercellular and does not produce haustoria. It becomes intracellular and then the host cell dies. Now abundant sporulation is present on the upper surface of leaves and these conidiophores are olivaceous in color, brown, olivaceous brown and yellow brown in color, one or two septa, short, unbranched, geniculate and arise in clusters. The conidia are subhyaline or pale yellow. That means they are almost transparent but not transparent. Obclavate, often curved with 3 to 12 septa. 35 in the size of the septa is 35 into 110 into 2.5 to 5.4 micrometer in size. Now they are roundish with distinctly truncate base and subacute tip. The perfect stage is producing perithecia and they are globose, papulate, ostiole and the esci formed in this are cylindrical to clavate. They contain 8 ascospores. Now the ascospores are hyaline, that means they are almost transparent, slightly curved, two-celled only, and the apical cell is larger than the lower cell. That was Personata ardicola. Then we have Cercospora personata. The second is the difference between the two is here the mycelium is intercellular. The conidiophores are long, continuous, 1 to 2 septa, arise in clusters, geniculate and olive brown in color. The conidia are cylindrical to obclavate, short and their measurement is 18 to 16 into 6 to 10 micrometer and they are hyaline to olive brown and usually they are straight but curved slightly with 1 to 9 septa. They are not constricted but 3 to 4 septa are there. Now here it is again the perfect stage is perithecia with ascostomata and the esci here are cylindrical to ovate and 8 ascospores are present. The ascospores are two-celled and they are constricted at septum and hyaline. Now the favorable conditions for this disease is high relative humidity for 3 days, Low temperature 20 degrees centigrade with dew on the low leaf surface. Heavy dose of nitrogen and phosphorus fertilizer and deficiency of magnesium in the soil. Now the disease cycle, the pathogen is surviving on the infected plant debris through conidia, dormant mycelium and perithecia. The groundnut plants also harbor the pathogen, and primary infection is through ascospores or conidia, and the secondary is through blown conidia. Rain splashes are also helpful in spreading. For the management, we have to destroy the infected plant debris. We have to eradicate the groundnut plants which are there present. We have to keep the weeds under control, and seed treatment with carbendazim or thyram at 2 grams per kg, or spray, and we spray carbendazim at 500 gram or mancozeb 2 kilogram or chlorothalonil 2 kg per hectare, and we repeat it if required after 15 days. Moderately resistant varieties are very helpful in managing the disease. Now, another disease which we have is wilt. It's a minor disease, and the germinating seeds are attacked by the fungus Fusarium pathogen before emergent shock. Now, there is tissue disintegration, and the sporulating mycelium often covers the seedling. The damping of symptoms are there, and brown to dark brown water-soaked sunken lesions are seen on the hypocotyle. They further encircle the stem and extend above the soil level. The roots are also attacked, especially the apical portion of the root, and ultimately the seedling becomes yellow and wilted. The leaves turn grayish-green and plant dries and die. Do you know why? Because the fusarium affects the internal xylem and phloem. So when the roots and stem are split open, internal vascular browning and discoloration is seen. Now this fungus is also associated with pod rot and is present there. So the only treatment is systemic fungicide like carbendazim at 2 grams per kg seed. So here you're seeing a plant which has wilt symptoms. There's drooping of leaves, yellowing of leaves, falling on the ground, etc. So students, I hope you have liked my lecture today, and if you have any query, please give it in the comment section. I will resolve it soon. This session was powered by Digital Version 2.0 of Jyoti Vidyapeeth Women's University. Namaste.